Hello dear students, today is lecture number 3 of chemical coordination and integration. Before we start with the details of today's lecture, I would quickly want to recap what we've done in the previous 2 lectures. So, this chapter began with a discussion on what is a gland. Any organ which secretes, that is produces or releases chemical substances, is called as a gland. After that, we went on to what is an enzyme, and we had a discussion that enzymes are defined as biocatalysts, that is, they are catalysts for biological reactions. If there are no enzymes, technically biological reactions do not take place. Then we moved on to what is the difference between an exocrine gland and an endocrine gland. So exocrine glands are glands with ducts, endocrine glands are ductless glands. Exocrine glands pour their secretion into the lumen of the duct, whereas endocrine glands pour their secretion directly into bloodstream. Exocrine gland have secretions that are called enzymes, endocrine glands have secretions that are called hormones. Exocrine glands have local effect, endocrine glands have effect on distant organs. A few examples of exocrine gland are salivary gland and liver, and a few examples of endocrine gland are pituitary and thyroid. We also saw that pancreas is a special kind of gland because it is a heterocrine gland. So as far as the pancreas is concerned, if you take a cut section of the pancreas and you see it under the microscope, this is how it looks. And then we saw that pancreas have exocrine cells which will pour enzymes, and they will pour the enzymes into the duct. At the same time, they also have endocrine cells which are called as the islets of Langerhans, which will pour their hormones into the blood capillaries. So pancreas is exocrine as well as endocrine, and hence pancreas is called as a heterocrine gland. Then we moved on to what are hormones. So hormones are basically chemical messengers produced by endocrine glands, released directly into the bloodstream, having effect on distant target organs, thus helping to maintain homeostasis. Next point was regarding the 12 properties of hormones, and the 12 properties of hormones are that hormones are chemical messengers produced by endocrine glands, released directly into the bloodstream. They control the functioning of distant specific target organs. Chemically, most of them are proteins. Once again, all of them are not proteins. They can be amines, they can be fats when they are called steroids. Most of the time, however, they are proteins. Hormones are produced in very small quantity. Even though hormones are produced in very small quantity, a slight increase or a slight decrease can lead to disease. Their action is slow and lingering or long lasting as compared to neural coordination or nervous coordination, which is extremely fast. Hormones are water soluble so that they can be easily transported by blood. They have low molecular weight. More importantly, hormones are destroyed and excreted as soon as their work is over, and hormones help in maintaining homeostasis. So what is going to be a gland? What is an exocrine gland? What is an endocrine gland? What are going to be hormones, and what are the 12 properties of hormones? This was the definition of hormones that we saw, which was given in the NCERT. Hormones are non-nutrient chemicals which act as intercellular messengers and are produced in trace amounts. So this is where the first lecture on chemical coordination had ended, and the second lecture began with me explaining to you that these are the various possible endocrine glands that you may see in the human body. So there are glands like hypothalamus, pituitary, pineal, thyroid, thymus, pancreas, adrenal glands, and the gonads, testes in males and ovaries in females. So these were all the endocrine glands which I told you we will be studying in this chapter. Before diving into pituitary in the last lecture, I did a long list of words which you can see on the screen right now. These were the words which we discussed before we moved on to the pituitary gland, and I quickly want to revise each word before we move on to pituitary. Inter means in between, intra or endo means inside, exo, epi, peri means outside, exo means outside, epi means outside and above, peri means outside and surrounding. Ventral is the side of the organism which is facing the ground. In the case of humans, it is forwards. Dorsal is the side of the organism facing the sky. In the case of humans, it can also be backwards. So we saw what is ventral and what is dorsal. Median, medial and lateral. So median is on the midline, medial is towards the midline, lateral is away from the midline. Tubercle basically means elevation. Fossa is going to be a depression. Sulcus is going to be a groove. Sinuses are going to be closed cavities. Chroma is color. Phil is to love. Phobe is to hate. Proximal means closer to. Distal means away from. So that is the entire list of words which we had discussed before starting pituitary in the last lecture. Then we started off with the pituitary gland, that in this diagram, this is the frontal bone, this is the parietal bone, this is the occipital bone, there is a temporal bone not visible here, this is going to be the ethmoid bone, and this here is the sphenoid bone, and this is where you can see the pituitary gland. So we had seen that the pituitary gland is going to be about 10 by 13 by 6 millimeters pea size, and it is going to be about 0.5 to 0.6 grams in weight. It is a small reddish brown colored gland. Pituitary gland is hanging from the ventral side of the brain, which is going to be called as the hypothalamus, via this dundee or stalk, which is going to be called as the hypophyseal stalk, also called as the infundibulum. So we had seen this. I had also explained to you that what you see in this diagram is going to be a horse's saddle from Turkey. So it's called as a Turkish saddle or sella turcica. This was important for us to know because the pituitary gland lies in this fossa of the sphenoid bone, and this fossa of the sphenoid bone is called sella turcica or hypophyseal fossa, which resembles the saddle of a Turkish horse, and that is why it is also called Turkish saddle or sella turcica. Now, if you were to magnify this, you can see that the pituitary gland is hanging from the hypothalamus with this funnel shaped structure, which is called as the infundibulum. So this structure here is going to be called as the infundibulum. So we have seen what is the position of the pituitary gland. Position of the pituitary gland is that it is located on the ventral side of the brain, hanging from the hypothalamus. It lies in a small cavity in the sphenoid bone of the cranium called as the hypophyseal fossa or the sella turcica. Externally, the pituitary gland is small, oval, reddish grey colored gland. It is going to be roughly about 10 by 13 by 6 millimeters pea size. Weight is about 0.5 to 0.6 grams, and it is attached to the hypothalamus by a funnel shaped stalk called as the infundibulum or the hypophyseal stalk. Here I would want to point out that pituitary gland is the smallest endocrine gland in the entire body. So in an adult, the smallest endocrine gland is going to be a pituitary gland. In fact, even in children, the smallest endocrine gland is the pituitary gland. So keep this point in mind. After that, we saw that male gametes and female gametes combine together to form something called as a zygote, which forms something called as an embryo. And we saw that the cells of these embryo are classified into the ectoderm, the endoderm, and the mesoderm. We did this because we wanted to have an idea about the embryology of the pituitary gland. So we saw that in this cross section of a human baby, this is where his brain is going to develop, which develops from the neural ectoderm. This is where his nasal cavity and oral cavity will develop. They will develop from the oral ectoderm, and somewhere here you will have the pituitary gland. So basically, if I was to magnify the blue box that you see, there is the upper part, which is the neural ectoderm, and there is a lower part, which is the oral ectoderm. The neural ectoderm moves downwards and the oral ectoderm moves upwards. So the oral ectoderm is moving upwards, the neural ectoderm is moving downwards. This upward extension of the oral ectoderm is also called as Rathke's pouch. This will continue, and as it continues, the oral ectoderm loses its connection with the oral side. However, the neural ectoderm continues downwards and maintains its connection with the neural ectoderm, the upper part. So this part forms the anterior side of the pituitary, this part forms the posterior side of the pituitary. So neural ectoderm moving downwards, oral ectoderm moving upwards, neural ectoderm continues to move downwards, oral ectoderm continues to move upwards. Oral ectoderm will eventually lose connection with its oral side and get attached to the posterior side. So now what you have here is the pituitary gland. This is the anterior part developing from the oral ectoderm, and this is the posterior part developing from the neural ectoderm. This entire thing is how the pituitary gland develops as far as its embryology is concerned. Also bear in mind that this is going to be called as Rathke's pouch. So embryological division of the pituitary gland can be the one which develops from the mouth, which is called as the adenohypophysis, and one which develops from the brain, which is going to be called as the neurohypophysis. Adenohypophysis develops from the oral ectoderm as a pouch called Rathke's pouch, and neurohypophysis develops as a downward growth from the neural ectoderm. This is the embryological classification of the pituitary gland. However, NCERT says that this is the anatomical division of the pituitary gland. So we will stick with that. So anatomically, adenohypophysis and neurohypophysis. Now what are the other various parts? This part, as we discussed before, is called as infundibulum. This part in the adenohypophysis is called pars distalis. This part in the neurohypophysis is called as pars nervosa, and this part in between, the one which you see here, this part here is going to be called as the pars intermedia. Pars intermedia in humans is almost merged with the pars distalis, and together these two are going to be called as the adenohypophysis. This area is going to be the adenohypophysis developing from the oral ectoderm. This part is developing from the neural ectoderm. Okay. Pars distalis, just this part, not this part, is called as the anterior pituitary. So this is the anterior pituitary. So anterior pituitary plus pars intermedia is going to be adenohypophysis, and pars nervosa is the entire neurohypophysis, which can also be called posterior pituitary. So I hope this clarifies what is the part of adenohypophysis and what are the parts of neurohypophysis. Blood supply of the pituitary gland. Before diving into blood supply, we spoke about the heart and the blood vessel carrying blood away from the heart, which will be called as the artery, which divides into smaller blood vessels called as the arterioles, which will then form the capillaries. And here we saw that capillary is the only part where material can go outside from the blood into the tissue fluid, and from the tissue fluid to the various cells. Whatever are going to be the waste products produced by the cells will go into the tissue fluid, and from there they can enter again into the blood capillary. So here the important point to understand is that exchange of material cannot happen in an artery, cannot happen in an arteriole, will only happen at the level of a capillary. The capillary is rejoined to form something called as venules, which then form bigger vessels called as veins, which will carry the blood back again to the heart. This picture that you see on the screen explains to you the basic fundamental properties of circulation, which is that arteries carry blood away from the heart, divide into arterioles, forms capillaries, which are the only blood vessels where exchange of material can take place, which reunite to form the venules, which will then join to form the vein, which will then come back to the heart. Okay. So here we now see the blood supply of the pituitary gland. The posterior pituitary is quite simple in its blood supply, which is exactly how we discussed right now, that there will be an artery divides from arterioles, which will form the capillaries, which will form the venules, and then there will be the veins. However, the anterior pituitary is a little complicated. As far as anterior pituitary is concerned, you will have the artery, which divides to form the arterioles, they form the capillaries, they form the venules. Now these venules will form veins, but those veins do not go to the heart. Instead, these veins will go from the hypothalamus to the hypophysis, the anterior pituitary. And here again they will divide into capillaries and venules, which will reunite to form veins, and now these veins will go back to heart. So as you can see, the anterior pituitary has a complicated blood supply because of these vessels, which instead of returning back to the heart are moving from the hypothalamus to the hypophysis. Hence these are simply called as the hypothalamo-hypophyseal portal veins. The word porta means to join, and here the portal veins are joining the hypothalamus with the hypophysis. So this shows you the entire blood supply of the pituitary gland, the anterior pituitary having the hypothalamo-hypophyseal portal veins, and the posterior pituitary showing normal blood supply as discussed before. Then the last part of the last lecture was histology of the pituitary gland, which I told you will be continued in today's lecture. So I hope that everybody is extremely clear on the previous two lectures. If there are any concepts which you have forgotten, which you do not remember or recollect, were finding it difficult to imagine when I was doing this quick revision, I suggest that you go back and quickly have a look at the previous two lectures before moving on to this one. Today's lecture begins now. Again, before we start today's lecture, there is a list of words that we need to know, which you will see on your screen right now. So you have pre or pro, post, soma, trough, mammary glands, lactation, gonads, supra, infra. So what do all these words mean? Let's have a look at them one by one. Pre or pro means before and post means after. So you must have heard these words quite often, that preschool, pre-primary. So basically that means before school or before the primary section. Post means after. So pre is something you use to denote कि कुछ पहले हो रहा है, and post is something to denote कि कुछ बाद में हो रहा है. Soma simply means body. Trough means to stimulate. So anything which is going to stimulate something, you can use the word trough. Mammary glands, of course, as you know, are organs which are going to produce milk. So an entire category of animals called as mammals will have mammary glands which can produce milk. The process by which mammary glands produce milk is called lactation. So mammary glands produce milk. Process by which mammary glands produce milk is going to be called as lactation. Gonads are organs which are going to produce the gamete. So gonads are reproductive organs, but all reproductive organs are not gonads. So for example, a human female will have an ovary where the ovum is going to be produced. She will also have a uterus or a womb where the baby will grow. She will also have mammary glands which are going to produce milk. Now all of these are reproductive organs, but all of them cannot be called gonads. So what are gonads? The organs which produce the gamete are called as gonads. So in male, the gonads are going to be the testes which produce the sperm, and in female, the gonads are going to be the ovaries which are going to produce the ovum. So I hope this point about what is a gonad and what are reproductive organs is clear. All gonads are reproductive organs, but all reproductive organs are not gonads. Okay. Lastly, you have supra and infra. Supra basically means above and infra basically means below. So quick recap of these words. Pre or pro means before, post means after, soma means body, trough means to stimulate. Mammary glands are those glands that are going to produce milk in the case of mammals. Lactation is the process of production of milk by the mammary glands. Gonads are those reproductive organs which produce gametes. All gonads are reproductive organs, but all reproductive organs are not gonads. Supra means above and infra means below. What we need to discuss in today's lecture is histology of the pituitary gland, and histology of the pituitary gland means how does the pituitary gland appear, how do the cells appear when we see them under the compound microscope. Now all of you from school are aware of this fact that the compound microscope is basically using light to magnify an image. So it uses light and it uses lenses. Whatever you wish to see is put here in a slide. There is a light source here. So once you put on this switch, this will illuminate and the light from here will pass through this hole, whatever specimen you've placed here, and there is a lens here and there is a lens here. So these two lenses, by the principle of optics, will amplify or magnify your image. This lens which is close to the object is called as the objective, and this lens which is close to the eye, which is where you will see into the microscope, is called as the eyepiece. Everybody must have studied this in your school level physics, that this is your object that you wish to see. This is the first convex lens which will magnify this object here, and this is the second convex lens which will magnify this object here. This object appears to be this big when you see it through the microscope. The lens close to the object is called as the objective. The lens close to the eye is going to be called as the eyepiece. So all of these things you are already aware of because you must have done them in school. Let us now take this compound microscope and now let's take a sample. So let's take a drop of blood onto a slide and take that slide and put it in our microscope and try to see what will appear through the microscope. So here's what you will see. When you look into the microscope, after having placed a drop of blood onto a slide and you are putting it in the stage of the microscope, this is what you will see. nothing. It will just appear to be a bright box in which you will not be able to see anything at all. So now, even though you've put a drop of blood, why are you not able to see anything? So that brings us to the concept that what specimen can you actually see in the microscope? So for you to see something in the microscope, there are two main conditions that have to be fulfilled. The first one is that it should be transparent. So whatever you put here has to be transparent, because if it is not transparent, the light from here will not pass through these lenses only. And as we just saw or revised, if the light does not pass from here, there is no ray diagram only. For ray diagram, you require rays. So if those rays only don't go into the microscope, what are you going to see here? So it has to be transparent. Whatever you place here has to be transparent. And the second thing is, it should have some color. So this is a really important point, because if you put something here which is transparent, as the light passes through, if it is transparent, what you will only see here is the white bright light, because it is transparent. But if it has color, the white light which comes from here will take up that color, and that colorful image will be seen here. So it should fulfill two criteria. Whatever you place in the compound microscope should fulfill two criteria. Criteria number one, that it should be transparent, and criteria number two, that it should have color. Now the first criteria, it should be transparent, is easy to fulfill simply because most of the cells are so small that they are transparent. They will allow light to pass. However, the second point, that it should have color, is something we need to take under consideration, because most of the cells do not have color. Most of the cells which you would want to place here do not have color. So now what do you do if you want to see something under the microscope? So let's say that here is the specimen of your cell which you wish to see under the microscope. You know it is transparent, but now you have decided that I need to color it, otherwise I will not be able to see it under the microscope. So अभी तुम क्या सोच रहे हैं? चलो होली खेलते हैं. Let's color this cell completely. But the thing is that you just cannot take any random pigment or any random chemical which has color and place it on the cell. Here's what is done. You take this cell and you have special commercially available colors like eosin and methylene blue. As you can see in the diagram, eosin is having this purplish pinkish tinge, whereas methylene blue, as the name suggests, is a blue color. Now these are the colors which are commonly used to stain cells when you want to see them under the microscope. The important property of these colors is not that they are pink and blue. The important property is that eosin is acidic and methylene blue is basic. Now why are these acidic and basic properties of these colors so important when you want to stain or when you want to color this cell? The reason for that is that as far as the cell is concerned, there are some parts which are going to be acidic and there are some parts which are going to be basic. If you take any color which is neutral and you throw it on the cell will simply not absorb it, because the cell has acidic areas and basic areas. But if you take colors like these, eosin and methylene blue, which are acidic as well as basic, so when you take eosin and you put it on the cell, all the parts of the cell which are going to be basic will love this acidic color and absorb it. So do not get confused here. Mark my words, whichever are the parts of this cell which are basic will readily uptake the acidic color. So it doesn't matter whether the color was pink, orange, violet, indigo, blue. What is important is that the color was acidic, so whatever is basic in the cell will immediately absorb it. So basic parts of a cell will look pink because they absorbed the acidic color. These basic parts can be called acidophilic. So now recollect, the word phil means to love. So acidophilic basically means acid se प्यार करने वाला. Again remember that acidophilic does not mean कि खुद acidic है. Acidophilic means कि basic है और acid से प्यार करता है. So all the parts of these cell which are basic, when you put these two colors on it, the acidic color will be absorbed by those parts. So the basic parts of the cell will absorb the acidic color and look pink. Similarly, if you were to put the basic color, all the acidic parts will absorb it. So the acidic parts of the cell love the basic color and will appear blue. So they are called basophilic. So cell will have acidophilic regions, a cell will have basophilic regions. The acidophilic regions are basic and love acid, and the basophilic regions are acidic and love the basic color. Okay. So now you can see that the nucleus is completely blue. So take a minute here and think about it. It is blue in color, which means that it has absorbed methylene blue. Methylene blue is basic. Basic colors will move towards the acidic part. So why is the nucleus blue? Because it contains DNA, deoxyribonucleic acid. All nuclei contain DNA. DNA is acid, hence it picked up the basic color. So I hope that from now on, whenever you see a cell in the microscope, the nucleus will always appear blue, because it will always absorb the basic blue color, because nucleus contains acid, deoxyribonucleic acid. Clear? Okay. The other parts of the cell may look pink or may look blue depending on the cell. So if the cell has acidic cytoplasm, it will look blue. In this case, the cell has basic cytoplasm, so I have shown it as pink. This is the principle of staining. This is important everywhere in biology. So I hope that this concept is now drilled into your head, that if you want to see something in a microscope, it has to be transparent and it should have color. Most cells are going to be transparent, but most cells do not have color. So we have to color them. To color them, we just cannot use any ordinary pigment or chemical. We have to use specialized chemicals which have different pH or different acid base value. The normal color that we use in biochemistry or in histology are eosin and methylene blue. Eosin is acidic and pinkish, methylene blue is basic and bluish. All the acidic parts of the cell will be basophilic and take blue color, and all the basic parts of the cell will be acidophilic and take up the pink color. Are we ekdam clear on this? So if you now have this concept under control, we will take the drop of blood, but to this drop of blood, we will add a drop of eosin and a drop of methylene blue. And now we will take the slide and we will place it under the microscope, and now here is what you will see. All the different cells like the RBCs, the WBCs and the platelets are clearly visible. Again here, notice how the nucleus of the WBCs is appearing blue. The cytoplasm may be of different color, and that doesn't matter here. What matters more is that the nucleus is going to appear blue. The nucleus is appearing blue because it contains acid, which is basophilic. The acid loves basic, and that is why they appear blue. Also, you will notice that the RBCs have this white area in between. This is not their nucleus. RBC does not contain nucleus at all. They are enucleated, and hence you cannot see any blue nuclei in the RBCs. So today we wanted to do histology of the pituitary gland, which is seeing the pituitary gland in the microscope. Before we do that, it was essential for you to understand the concept of seeing in the microscope. I have spent so much time explaining this because this is a basic concept which will be important in your future in all the lab experiments that you do, and even when you go into medicine, these same concepts are going to apply, and that is the reason why we spend so much time with this. So for the last time, acidic parts are basophilic and stain blue, basic parts are acidophilic and stain pink. Clear? Super. Let's now move on to the histology of the pituitary gland. So finally we can start today's actual topic about the pituitary gland, which is histology of the pituitary gland. Let's take the pituitary gland, let's cut a small part of the adenohypophysis and place it under the microscope, and let's see what will appear there. Okay, so here is what you are going to see. In the microscope, you will notice that the pituitary gland, about 50%, comprises of these uncolored small size cells. Now they are uncolored, or they did not pick up any stain because they are very small. Their cytoplasm is neutral, so neither acidic nor basic, so it's not going to pick up any color. And Because they are very small, the nucleus will also pick up very little amount of the blue color. So they almost appear colorless in the microscope. These cells are called chromophobes. Chromo meaning color and phobe meaning hating. So they do not absorb any color and are called chromophobes. Along with the chromophobes, you are going to see these big cells which have either pink or blue color. That means that their cytoplasm had enough amounts of acidic or basic components which have picked up either the basic or the acidic color. Now these cells which have picked up the stain, have picked up the color, are called chromophils, color loving, and they will either appear pink or blue. So now the pink cells have taken up the acidic color, they are acid loving, that means they themselves were basic, and the blue cells are going to be basic loving, that means they have picked up the blue color, meaning that they themselves were acidic. So they can be called as acidophils or they can be called as basophils. Acidophils loving acid, basophils loving basic. So this is what the pituitary gland appears under the microscope, comprising of the chromophobes, which are about 50%, do not take up the stain, and the chromophils, which are going to be another 50% and will pick up the stain. The chromophils will be about 40% acidophils, which are pinkish, another 10% basophils, which are bluish. So this is what you see. Now these cells, the chromophobes, do not produce any hormones, whereas these cells, which are the chromophils, are going to produce hormones. So the chromophils, acidophils and basophils produce hormones, and that is what we are going to see in the subsequent slide. Adenohypophysis comprises of two parts, the pars distalis and the pars intermedia, which we had seen in the previous lecture. The pars distalis has cells which secrete hormone. The pars intermedia in humans is very small and is almost merged with the pars distalis. The pars distalis has two main types of cells. You have the chromophils and the chromophobes, about 50-50%, and the chromophils are further classified as acidophils and basophils. Acidophils are pinkish and basophils are bluish. So this is what we did in the previous slide. Now the pink cells and the blue cells are going to release hormones, and I am now going to tell you the hormones that are produced by each of these cells. Before we start that, today we are only discussing the names of the hormones. As the chapter progresses, and of course in the next lecture, we will see each hormone in detail. But as of today, I just want you to focus on their names and what is the simple meaning of their name as to what the hormone does. So acidophils produce two main hormones, which are growth hormone and prolactin. As the name suggests, growth hormone is a chemical messenger which will tell the parts of the body to grow, and prolactin has two words, pro and lactin. Pro means before and lactin means lactation. So prolactin is a hormone which comes before lactation, comes before a mammal, a female mammal, can produce milk with her mammary glands. So you have growth hormone and you have prolactin. Now the cells which produce growth hormone are pink, the cells which produce prolactin are also pink, but in the microscope you cannot distinguish that which pink cell is going to produce growth hormone and which pink cell is going to produce prolactin. They will look pink. However, the one which produces growth hormone does not produce prolactin, and the one which produces prolactin does not produce growth hormone. So they are separate cells as far as their functioning is concerned, but in the microscope you cannot distinguish them. Growth hormone is also called somatotrophic hormone, body stimulate hormone, somatotrophic hormone. The third name for growth hormone is somatotropin. So you take somatotrophic hormone, hormones are mostly protein, somatotrophic protein, somatotropin. Growth hormone, somatotrophic hormone, somatotropin. So I hope these three words are clear with everybody. So the acidophils which produce this are called somatotrophs. Somatotroph is the cell which produces somatotrophic hormone. Similarly, prolactin is called mammotrophic hormone, mammary gland को stimulate करने वाला hormone. So these cells, pink cells, which are going to produce this hormone are going to be called as mammotrophs. So in the pituitary gland, if you cut the adenohypophysis, you will have color loving, color hating. Loving cells are going to be of two types. They will either be acidophils or basophils. And now the acidophils are of two types, not seen in the microscope, but function wise they are of two types. They are going to be the somatotrophs and the mammotrophs. Somatotrophs produce growth hormone, somatotrophic hormone, somatotropin. Mammotrophs are going to produce the prolactin, mammotrophic hormone or mammotropin. So this was the acidophils. Now basophils produce thyroid stimulating hormone. Now I think the name is self-explanatory. Thyroid stimulating hormone, thyroid ko stimulate karne wala hormone. So that is what that hormone is going to do. The next one is going to be called as adrenocorticotrophic hormone. So now from this word you can see that's a hormone that is going to stimulate, and what will it stimulate? Adrenocortico. Now what is adrenocortico? So for that I want you to have a look at this picture. So this is a simplified version of the human body, and you all know that we have these two organs which are called kidneys. If I was to take this kidney and if I was to magnify it here, this is how it looks. Now the Latin name for kidneys is renes, and this organ here is adjacent to renes, adjacent to renes. So this organ here is called as the adrenal gland. So you have the renal kidney and the adrenal. This part here is the adrenal gland. Now if you were to cut this adrenal gland, if you were to cut it, so if this was the adrenal gland and you were to cut it, this is how it appears. It clearly has an outer part which you can see here and an inner part which you can see here. The outer part is called as the adrenal cortex and the inner part is called as the adrenal medulla. So you have the cortex and you have the medulla. Adrenal cortex and adrenal medulla. The hormone which we just saw the name of, adrenocorticotrophic hormone, adrenal gland के cortex को stimulate करने वाला hormone, which means that this adrenocorticotrophic hormone will stimulate only the adrenal cortex, only this part is going to get stimulated. Once this part gets stimulated, what it is going to do, which part it is going to affect, what are the things that this part will do is a discussion for the next lecture. But right now I just want you to know adrenocorticotrophic hormone stimulates this, the adrenal cortex. So basophils produce thyroid stimulating hormone, basophils produce adrenocorticotrophic hormone. They also produce gonadotrophic hormone. So we just saw that gonads are organs which produce gametes. So that hormone that stimulates the gonads ke gamete banao will be gonadotrophic hormone. So basophils have thyroid stimulating hormone, adrenocorticotrophic hormone, gonadotrophic hormone. The cells, the basophils which produce thyroid stimulating hormone are now called thyrotrophs, which produce the adrenocorticotrophic hormone are called corticotrophs, and those which produce gonadotrophic hormone are called gonadotrophs. So in the microscope, all basophils look blue, but some of them produce thyroid stimulating hormone called thyrotrophs, some of them produce adrenocorticotrophic hormone called corticotrophs, some of them produce gonadotrophic hormone and are called gonadotrophs. Is this clear? So we have the acidophils and the basophils. Acidophils produce growth hormone, prolactin, somatotroph, mammotroph. Basophils produce thyroid stimulating hormone, adrenocorticotrophic hormone, gonadotrophic hormone. The thyrotrophs, corticotrophs, gonadotrophs. Thyroid stimulating hormone is also called thyrotrophic hormone, and as we saw previously can be condensed to thyrotropin. Adrenocorticotrophic hormone can also be called corticotrophic hormone, can be condensed to corticotropin. Gonadotrophic hormone can be condensed to gonadotropin. So these are the three cells of the basophil variety and their respective hormones. Also, pars intermedia produces a hormone called MSH, melanocyte stimulating hormone. So if I was to quickly summarize what is going to be the histology of the adenohypophysis, how adenohypophysis looks under the microscope, we will see that there are two main parts. There is a pars distalis and there is a pars intermedia. The pars intermedia is highly reduced and produces only one hormone called as MSH, melanocyte stimulating hormone. The pars distalis has two types of cells, those which are going to be color loving and those which are color hating. The color hating cells are going to be called as chromophobes. The color loving cells are going to be called chromophils. These do not produce any hormones. These chromophils will produce hormones. Chromophils, the color loving cells, are going to be of two types. Either they are basic and love acid, acidophilic, or they are going to be acidic and love basic, basophils. So you have acidophils and basophils. Acidophils are of two types. In the microscope you cannot distinguish between the two, but they are of two types: somatotrophs, mammotrophs. Somatotrophs produce growth hormone, somatotrophic hormone, somatotropin. Mammotrophs produce prolactin, mammotrophic hormone, mammotropin. Basophils are going to be of three types. They are thyrotrophs, corticotrophs, gonadotrophs. Thyrotroph produce thyroid stimulating hormone, thyrotrophic hormone, thyrotropin. Corticotrophs produce adrenocorticotrophic hormone, corticotrophic hormone or corticotropin. Gonadotrophs produces gonadotrophic hormone or gonadotropin. This completely finishes off what you see under the microscope as far as the anterior pituitary is concerned. In the next lecture, we are going to be discussing what do we see when we take the posterior pituitary under consideration. So this concludes today's lecture on histology. We have not finished the entire histology of the pituitary gland. We have only seen the histology of the anterior pituitary. How does the posterior pituitary look under the microscope is still left to be done. We will stop here today because I want everybody to have clearly grasped and digested the idea of how does a compound microscope work, and also this entire chart which you see on your screen right now as to what are the hormones produced by the anterior pituitary. In the next lecture, we shall continue with what are the hormones which are produced by the posterior pituitary and how does the posterior pituitary look under the compound microscope. So till next lecture, goodbye, have a great day.